We're going to get started on the last topic of the course, which is differential expression analysis. So this is super common for bulk RNA-seq. Pretty much everything you do with bulk RNA-seq is take two populations, do RNA-seq, and then do differential expression between them. So there's many methods that have been designed for bulk RNA-seq, such as DSeq, EDGAR, and lemma-voom, which are specifically tailored for datasets where you have very small number of samples and very high coverage of the genes in each sample. Whereas in single-cell RNA-seq, we have the opposite. In single-cell RNA-seq, and it's also defined experimental groups. So EDGAR, DSeq, they assume you have some experimental setup where you have condition A and condition B. You know which samples belong to each condition, and you're just going to test the difference between them. Whereas in single-cell RNA-seq, we typically have a large number of samples, but relatively low coverage of the genes in each sample. And often we don't have clearly defined experimental conditions. Often we're interested in actually the heterogeneity within each condition rather than just testing group A versus group B. And in general, if you can do group A versus group B, bulk RNA-seq will give you better sensitivity and resolution of differentially expressed genes between group A and group B. If that's all you're interested in, do bulk RNA-seq. So for single-cell RNA-seq, we have different in that we're not just interested in group A versus group B. We have some heterogeneity going on. And often we're not necessarily just interested in the difference in the mean expression. We might be interested in different variances. So a gene might be more noisy in one condition versus the other condition. It might be very stable across all of the cells in the other condition, which we can only test by having a large number of cells and testing for a difference in distribution. So there's two main approaches for comparing distributions. Either you can use some sort of non-parametric test where your test is independent of what that distribution is, and you're just testing whether there is some sort of difference between those distributions, but you're not necessarily going to tell you what exactly that difference is. Or you can use a statistical model of the distribution, fit the distribution, that model, to the distribution you see in each group, and then test for differences in the specific parameters of that model across the different groups. So there's three main models of single-cell RNA-seq data for testing the different distributions. So the first one is the negative binomial model, which is what's used by bulk RNA-seq. They use a standard negative binomial as well. So this is what it looks like. These are the parameters in R that you can run. So you've got the mean, and then the size parameter affects the variance. So if you have a very large size value, you have a small variance, whereas if you have a small size value, you have a larger variance. And the negative binomial distribution. This is generally used for UMI tag data. It generally fits that kind of data quite well. But it doesn't usually fit full transcript data, because full transcript data, we often have some group of cells where there's relatively high expression and we have high read counts, but then for a large number of cells, we have just 0 read counts. So for that, most methods will use a 0 inflated negative binomial model. So this is just a negative binomial model, again, only for some percentage of the cells you set the value to be 0 regardless of what it actually would have been under a negative binomial. And there's many different models out there that use different ways of deciding what D will be in this case. So it might be fit to each gene separately, or it might be dependent on the mean expression in some way or another. And different methods will use different ones. So again, you can calculate the mean and the variance using these parameters to fit each of the parameters. But because we have 3 parameters now, if you want to fit the model to each gene, you need a third characteristic in order to fit all 3 parameters correctly. And then the third model that's specific to single-cell RNA-seq, which is actually based on a mechanistic model of how transcription occurs, is the Poisson beta distribution. So here we've got 3 parameters, alpha, beta, and gamma, which I've just called A, B, and G for simplicity. And here we've got a beta distribution for A and B, where A is the rate that a promoter is being turned on. B is the rate that promoter is being turned off. And that will determine, using a beta distribution, lambda, which is the proportion of the time your promoter is being turned on. And then it assumes you have some amount of read counts that's proportional to this lambda times gamma, which I seem to have forgotten. So you would then multiply lambda by gamma to be the rate of transcript production from that locus while it is active. And again, you can calculate the mean and variance for the distribution, although calculating the specific distribution is not analytically tractable. So it's generally much harder to fit this kind of model to your data than the negative binomial or zero-inflated negative binomial. But it has this benefit of being sort of interpretable, and there's a lot of evidence from actual experiments showing that transcription should follow a negative binomial distribution. So I'll give you a few minutes to look at these different models and change the parameters to see how different or similar you can get each one to be to each other. So we have about 5 minutes to play with the models, and then we'll move on to the next section.